In today's lecture, we learn about bioavailability and bioequivalence studies under the course of biopharmaceutics and pharmacokinetics. So let's start. So what is bioavailability? Bioavailability is defined as the rate and extent of absorption of unchanged drug from its dosage form. So it is the availability of the drug in systemic circulation. Then absolute bioavailability is the comparison of the drug taken by orally with that of IV administration. Then the relative bioavailability, whenever we are comparing the bioavailability of one drug with that another drug, then it is called relative bioavailability. So we will discuss absolute and relative bioavailability in the upcoming lectures. So let's learn the bioequivalence. So first of all, we need to learn that what is equivalence. It is a comparative term in which one drug product is similar to another with respect to chemical or molecular structure or specific characters or therapeutic action or different set of standards. So what is bioequivalence? If different doses from has the same drug, same dose, same bioavailability and same rate and extent of absorption and same Tmax, same AUC, and all of these criteria would be similar for two different drugs, then we called they are bioequivalent. So what is bioequivalence studies? These are the studies to identify the bioequivalent products, and another term is therapeutic equivalence. So same clinical effect, same safety. For example, propoxyphene hydrochloride versus pentazocine hydrochloride for the treatment of pain. Then we learn about pharmaceutical equivalence. So actually, what does it mean by pharmaceutical? Usually, when we prepare any doses from any API, it is called pharmaceutics. So it means usually they would be equivalent in term of their doses from. Okay, so they would have same active drug, same salt, star or chemical from, same doses from, and also in same identical in strength, concentration of route of administration, but they would differ in shape, release rate, packing and excipients. For example, chlordiazepoxide hydrochlorate 5 mg capsule, etc. So this is the overall pharmaceutical equivalence. So here is a capsule and here is another capsule. Both are of same drug. They are also of same doses from like capsule, and they are also identical in their concentration like 5 mg and 5 mg. But they may differ in their shapes, release rate, packing, or excipients. Then another term is pharmaceutical alternative. So this means they would alter in their pharmaceutical properties like different salt or star, different doses from, different strength. Okay, but same API. For example, diclofenac sodium and diclofenac potassium. Okay, so these are pharmaceutically alternative. Then tetracycline hydrochloride, there is tetracycline phosphate. Then another is quinidine sulfate, another is quinidine sulfate capsule. Okay, here is tablet and here is capsule. So for any drug to be pharmaceutically equivalent, they must be bioequivalent as well as therapeutic equivalent. So let's discuss the brand name, chemical name, generic name and drug product. So chemical name, it is the name of chemical structure of the drug. For example, acetyl salicylic acid. Then generic name, which is also called non-proprietary name. For example, name of any active drug like aspirin. Then brand name, it is also called trade name or proprietary name. So name of the drug product given by the manufacturer, for example Disprin. Then another one is drug product, it is the finished pharmaceutical dosage form like tablet, capsule or syrup. Now let's see different term which is used in bioequivalence or bioavailability studies. Then standard sample. So it is the sample in which we compare our test sample. So this is also called reference sample. It's innovator's product successfully proven and marketed, accepted by medical profession, generally high viability form. And uh, another is test sample, which is actually measured by us. Same API as innovator's drug, pharmaceutically equivalent, not proven yet successfully. Same procedure for manufacturing, same instructions and indications. Okay, so these are the test sample which usually we use for applying sample, and this is the standard with which we compare. Okay, so development of new drug molecule in the stage of new drug application in clinical trial. Here is development of generic products. When a patent on an innovator's drug expires, the generic product can be developed at cheaper economic rate. So this is the difference between brand and generic drug. There are other differences like branded drug are intended for new drug application, but generic drug is for abbreviated new drug application. So why abbreviated? Because for generic drug we usually don't perform preclinical or clinical trials. Then chemistry, these are similar. Then manufacturing controls also similar. Then labeling and testing is also similar for both branded and generic drug. But for branded drug, it's compulsory to perform preclinical trial like animal studies for safety and efficacy reason. But for generic drug, it is not required. Moreover, for branded drug, we usually perform clinical studies, pharmacokinetic studies, pharmacodynamic studies. Okay, but for generic drug, just you need to do is bioequivalence or bioequivalence studies. So as we have already learned that relative viability is the viability of drug product from another drug product as compared to the recognized standard. Okay, so this is the EUC of A and EUC of B. This is the relative bioavailability. Just for example, if we compare uh, any tablet like paracetamol tablet with its paracetamol solution, then this comparison would be relative bioavailability. And another one is absolute bioavailability. So whenever we are comparing any doses from with its IV doses from, then it is called absolute bioavailability. When the systemic availability of a drug administered orally is determined in comparison to its intravenous administration, then it is called absolute bioavailability. So it's denoted by F. Its determination is used to characterize the drug inherent absorption properties from the site. IV is selected as standard because the drug is administered directly into the systemic circulation and avoids absorption step. So here is a practice problem. Just you can see here the dose has been given, then the AUC has been given. All you need to do is to calculate the relative and absolute bioavailability. And for relative bioavailability, we have already learned that you have to just compare the tablet with the solution. Okay, so just compare the tablet with the solution, and the result is 1.04. And again, the absolute bioavailability, in absolute bioavailability, we just compare this with the IV route. So one question may arise that why absolute bioavailability is not always 100%. Okay, so to have a greater idea about this, first of all, we need to understand the basic physiology of our veins and hearts. So we are giving the drug by intravenous injection. Okay, so just think this is the intravenous, this is a vein. So actually, what is the direction of the vein? Whenever we are administering any drug through this vein will ultimately comes into where? Will ultimately comes into the heart. So this is the heart. Okay, the vein will ultimately come into the heart because this is the direction of the vein. Vein always moves from body to heart. So whenever you are applying any drug in this vein, it will come into heart, And we know already that from the heart, usually from the right ventricle, the blood uh, pass into the pulmonary system through the pulmonary artery. Okay, we already learned this in our physiology chapter. But whenever drug is passing the pulmonary system, it is undergoing metabolism. That's why uh, absolute viability by the IV route is not always 100%. It is true only if the API reaches arterial blood without loss. Drugs are generally administered by the IV route and have fast to cross the pulmonary circulation before gaining access to arterial blood. Lungs can be site of extensive fast pass and reduced drug availability. This is the case for prostaglandin and some amines and reduced drug availability. This is the case for prostaglandin and some amines. So let's discuss the requirement for viability and bioequivalence studies for investigational new drug and new drug applications to establish equivalence between early and late clinical trial formulations, formulation used in clinical trials and stability studies, then clinical trial formulation and to be marketed drug products, any other comparison if appropriate, then ANDA for generic drug product, change in components, composition or manufacturing process, change in dosage form. Okay, so these are the all cases when viability or bioequivalence studies are required. So here are the objects which are as like as those requirements. You can easily understand that by reading this. But there are some cases when bioequivalence studies are not required. Okay, this is also called biowaivers. Okay, biowaiver means those bioequivalence studies which is actually not important to perform. Okay, first of all, for parenteral solution. Okay, because for parenteral solution, absorption is already 100%. Okay, so same active substance with same concentration, same excipient. Then oral solution, they are already ready to dissolve, so no need for bioequivalence studies. Same API with same concentration, excipient not affecting the GI transition or absorption. Then gas. Okay, it is very difficult to understand the bioavailability pattern of gases like inhaler or meter dose inhaler or nebulizer. Then powder for reconstitution as solution. Then otic, ophthalmic or topical solutions, some active substance with same concentration and same excipients. Then inhalation products or nasal spray. For all of these products, it's impossible to perform the bioequivalence studies. So we have already read this. Chemical equivalence means it is indicated that two or more drug products that contain the same labeled chemical substance as an API in the same amount. So we have already discussed this earlier. So let's discuss different methods for assessing bioavailability. We know there are two types of methods for measuring the bioavailability in our body. First one is indirect method. We usually perform this by pharmacokinetic method. Okay, like it is widely used method, and drug concentration in blood or urine reflects the pharmacological response of drug. Okay, so we perform this by urine and blood analysis. Then another one is direct method, which is also called pharmacodynamic method. It is not widely used method, so no assumption and direct observation, like acute pharmacological response, clinical observations. So let's discuss the blood analysis. So in our other courses, we have already learned the area under the curve. Okay, we have all learned about this curve. In this curve, we can see there is a maximum concentration which is called Cmax. Okay, this is the Cmax, and the time when Cmax is achieved, it is called Tmax. Okay, so this is Tmax. We already learned about this. Okay, and the x-axis means time and y-axis means the concentration. So these are the pharmacokinetic parameter for blood concentration measurement of drug, like Cmax, Tmax, area under the curve. And then for urine analysis, this is also possible. So it's measured only in the concentration of unchanged drug in urine, non-invasive method. Metabolites may interfere analytical method. So let's see the graph. Here is the graph. This is as like as the dumbbell shaped area under the curve. But the main difference is here is the rate of excretion and here is the time. So the time when more elimination is found is the Tmax. Okay. So let's discuss the acute pharmacological response. In that response, we usually see the pharmacological impact of the drug. Okay. For example, developed test formulation, both of these drugs are having mydriasis. So which one is having better? First, we usually perform this by uh, direct intervention. You can see here the pharmacodynamic effects. So we can also perform by bronchodilator effect. We have already learned that, for example, how much our uh, liver has expanded. Okay, in our physiology course, we have learned about uh, tidal volume, vital capacity, expiratory reserve volume, inspiratory reserve volume, inspiratory capacity. Okay, uh, so these are used for whenever we are applying some drugs into the body and body's lungs are expanding, so we can easily measure the expansion, and from that we can easily find out the effect of the drug in the body. So here is this pyrometer which measures the lung capacity. Okay, so we have already discussed this earlier. This is the pharmacodynamic parameter and these are the drug examples. We have also discussed this earlier. So now let's see different bioequivalence studies. So there are usually four types of bioequivalence studies which are performed on healthy volunteers. So if a new product is intended to be substitute for approved medicinal products, then equivalence must be shown. To ensure the clinical performance of each product, bioequivalence studies are carried out to compare the viability of generic products with the brand products. So there are four types of bioequivalence studies, like completely randomized design, randomized block design, repeated measures or crossover design, and the last one is Latin square design. So we'll discuss this one by one, but we can simply understand what these designs actually means. First of all, completely randomized design. This is the randomized one. Then randomized block design. This is also randomized, but in that case, the objects or the volunteers are divided into different groups according to their it can be according to their age, it can be according to their gender or whatever else. Then repeated measures or crossover design. So in this case we usually administer one drug for one person at a time, then after the washout period we then again apply the doses from. And final one is Latin square design. We all also see different maths according to this design. So let's discuss this one by one. First of all, completely randomized design. We have already seen that it is easy to construct. These are the random numbers which is allocated among experimental subjects. It can accommodate any number of treatment and subjects. It's simple to analyze, but here are a number of disadvantages. Like it's suited for few treatment. All subjects must be homogeneous, and otherwise random error will occur. Then randomized block design. We have already shown that in this block design, we usually block means these are the blocks. Okay, so this is male block. Will administer the drug into the male, then this is a female block. Okay, among all the volunteers these are male, these are female, so I have just separated them and providing them different drugs. Okay, they can also be separated according to their age. Okay, so first subject are divided into homogeneous group called blocks. Okay, these are the blocks, and then treatment are assigned at random within blocks. Subjects are divided as blocks considering similar background characteristics. So main advantages are effective and systematic grouping gives more precise result, no need of maintaining equal sample size, and any number of treatment can be followed. Disadvantages are missing observations in block require more complex analysis. Then repeated methods or carryover design. So as the name suggests, repeated, it means will administer drug repeatedly for an interval period of time. It is a kind of randomized block design where same subject serves as block. Same subject utilized repeatedly for each study period to call as repeated measure design. Carryover effect of drugs are measured. Advantages include precise economic and can be performed with few subjects, useful in observing effects of treatment over times in the same subject at different points of time and in different subjects over a specified period of time. However, there are a number of disadvantages like carryover effect due to preceding treatment and washout period necessary for 10 elimination half-life. So our last design is Latin square design. As the name suggests, in that case, we usually perform or give the drug by forming any square. Okay, how? For example, this is drug A, this is B, this is C, this is D. Okay, we have just categorized them. Then this is person number one, this is person number two, this is person number three, this is person number four. Okay, so I'm just putting the value, and I will just administer drug A to person 1, then we'll have a concentration like 3. Then drug B to person 1, then it will have a concentration like 2.5. Okay, then this value will then give C to patient 1, then D patient 1, then again I will give patient A the drug 2. Okay, so these are the usual processes. We will perform Latin square design and will administer the drug randomly. All other above designs are continuous trial. However, in Latin square design, each subject receives each treatment during the experiment. It is two-factor design. Carryover effects are balanced. The main advantages are it minimizes the variability of plasma profiles and carryover effect. Small scale experiments can be carried out, possible to focus on formulation variables. However, number of disadvantages like long time study, more formulations, more complex studies, subject dropouts rates are high. So these are the in vitro dissolution studies that we have learned in the pharmaceutical technology. Just go through this. So these are different levels of IV-IVC, in vitro in vivo correlationship. There are three levels of correlation. Number one is uh, level A correlation. It means how much of your drug would be dissolved and how much of your drug to be absorbed. So this is a level A correlationship. Okay, which means a linear relationship between dissolution and absorption. Then level V, it means the in vitro dissolution time is compared with mean in vivo residence time. This is not more point to point correlation as like level A. Data can be used for QC standards. Okay, so it compared with mean residence time. Okay, and level C, it's single point correlation. For example, T50%, Tmax, Cmax. This level is useful only as guide for formulation development or quality control. Now, we can easily understand the design like Latin square design or any other design if we see a problem and associated solution. For example, in crossover study between drug A, drug B, drug C, drug D, drug E. Okay, so these are the patient 1, 2, 3, 4, 5. So these are the drug CPA, CPB. Okay, ABCD are the drug. Okay, so how many drugs, okay, how many drugs are here? So our k value is 5. Okay, degree of freedom, it means how many factors, these are the 5. And how many patients? So n value is equal to 5. Okay, our value of n is equal to 5 because 5 persons available, and k value is 5 because we have 5 drugs. Following is the data of cpmax of these 5 drugs obtained after in vivo bioequivalence studies. Determine their bioequivalence by ANOVA test, that whether these drugs are bioequivalent to reference drugs. Okay, so we need to find out whether they are bioequivalent. So just as like before, we'll have the total of this column, we'll have the total of this column, we have put this in this column. Okay, so just take a null hypothesis that all of them are equal, and our alternative hypothesis would be they are not equal, like at least 2 drugs cpmax are not same. It is alternative hypothesis. So let's perform the math. So level of significance is 5%. Then first of all we'll calculate the total sum of square. Just uh, square every each and every of them and divided by uh, n. It has not been shown, but you have to divide this by n. And another one is just calculate the total grand total sum square divided by nk. So 132 divided by k value is 5 and value is 5. Okay, so we'll perform this 132 square divided by k value 5 and value 5. Okay, just get the value. So our tss has already been found. Then it is treatment sum of square. So what is treatment sum of square? Just calculate the summation of them. Minus this is the same for both this. And if we remove this from here, then what will ultimately found, we'll have the error sum of square. Just tss minus treatment ss. Just we have got this. Now we have to calculate the f value. So how we will find this? So we have treatment sum of square, we have error sum of square, we have total sum of square. Now we have also the degree of freedom. How? For this analysis we usually minus 1 from this degree of freedom. For example, like you can see first degree of freedom, just k minus 5 and minus 5 is equal to 4. Okay, our first degree of freedom would be 4. Then it is error is in the formula, we calculate this. Then total would be 4 plus 24 is equal to 24. Then mean sum of square, mean sum of square would be this divided by degree of their individual degree of freedom, this divided by their individual degree of freedom. Okay, then f test, in f test you will just 19 divided by 2.88, will just 19 divided by 2.88. So this is the f result. Now we need to compare it with the critical value. So what is the critical value for this table? Okay, where degree of freedom is 4 and n value is 20. So 420, just keep it this in value, 4 and 20. Okay, so you can see here 4 and 20. So what is the value? This is 4, and our 20 would be around this way. Okay, so you can easily understand it would be like 2.8 something. Okay, so we have also put this here, this value is 2.87. So you can easily understand that. So from the critical value our value is higher. Okay, so it means our null hypothesis has been rejected. Okay, then we can also compare this with the mean value. Okay, for having the differences, just you can usually perform the math in that way. Just put the differences, then what will you do? We'll see the basic differences and we'll ultimately get the value of their equivalency test. So here we can see in conclusion we can have a difference between these. So these are all from our first chapter. If you have any confusion, please inform me in the comment section. Thank you.